I will say one pet peeve of mine that I don't know if I've ever expressed this. When people say it used to really bug me, now it's kind of whatever. But when people would say cannabinoids instead of cannabinoids, and I was like, and my
Mhm.
argument for
Same.
that is,
Let's talk
do
about it.
you ever say cannabis?
That's the
No,
same thing I
no one
say.
says cannabis. Everyone says cannabis. So why do we say cannabinoids? That's like one little pet peeve,
Oh my
and
God, I'm
I
I'm
had to
right
let
there
it
with you.
go.
I am right there with you, but because I'm around people so much that say it,
Mhm.
now I say both and I kind of go back and forth. All right, so Chad Ternes, my friend. I've never actually met you in person, but we've been online LinkedIn and TikTok buddies for a while now. And I think you do super cool stuff. I don't know what our initial like connection was, but I feel like it was like maybe tissue culture or something.
So this would have been almost 2 years ago now, and I was starting to actually do LinkedIn. Like, I had never done any type of real social media or anything like that up to this point, and I decided, let's just start with LinkedIn, and I decided I wanted to get more into cannabis again. Like, I had done a little bit of it as, like, formally as a job, but that was kinda short-lived, and ultimately decided I was working in patents, like, I wanna get back into cannabis, and you were somebody that I saw your name a lot when it came to LinkedIn, to be honest, like, on posts and comments and stuff like that. Dan Fowler was another one, seemed to be, like, a lot of followers, a lot of people listen to you guys, just some main people, if you will, in the industry. So it's like, why not? Let's just reach out, connect. And so, yeah, it's kind of met you via Zoom, Skype, and kind of just went from there. And honestly, just I love your vibe and the hair. I've always loved it from day one. And yeah, you're great. And so I like
Aw,
you.
I'm blushing. That's awesome. I am one of the louder voices, I guess, on LinkedIn. And it's mostly, I don't like do posts very often, but I do get
Mhm.
into conversations a lot. I have strong opinions and I want people to know them.
Yep.
But
Well,
yeah.
and you also bring the perspective of you're like a formerly trained scientist as well.
Hmm.
So, and I think back years, decades ago, whatever, most of the people in cannabis didn't necessarily have formal science backgrounds. And
Hmm.
so it's cool. We're just definitely starting to see more and more of that. But again, given the legal status of it for since forever, yeah, it's not the easiest thing to always work on. So,
It's not. And I also feel like when I'm having conversations online, like I don't like to stray off topic. So, like,
mhm,
if somebody makes a post and then I comment on it and then somebody says, "Oh, but yeah, blah, blah, blah," I'm like, "That's not what the post is about. We're not talking about that." And
right,
I'm very like science and like logic and things it's just so, and there are rules and these are,
right.
and I think it's, a touch of the tism maybe that I
Mhm,
think a lot of scientists have. And actually I think I post on your TikTok the other day. Oh no,
mhm.
was it on LinkedIn?
It was on LinkedIn,
It was
yeah.
on LinkedIn when you were saying like you were talking about how in lab settings and things like that, or what was it?
It's like, yeah, highly technical environments. It's like intelligence is kind of just a given and competence. And a lot of times it just comes down to kind of people skills, communication, leadership skills. And so issues in kind of tech environments and labs, whatever, it's rarely about the science or the actual work that's being done. It's more of kind of the dynamics of the team, the leader, things of that nature. Because a lot of stuff just goes unspoken, but it just kind of becomes this low level undercurrent, if you will. I think a lot of people can relate to that. Neurodivergence, I think you definitely find quite a few of us in the STEM fields, and we don't always have the best people skills, right?
We
So,
absolutely 100%. I don't do eye contact. I watch people's mouths, but it's like the same kind of like we're supposed to, like
mhm.
we should be
Yeah.
facing you when we're talking to you. And that's just kind of like a little, I don't know, trick to not look at people in the eyes.
Mhm.
And still do that social nicety thing that you're supposed to do, which I'm
Right.
also terrible at. Like, Hi, I hope this email finds you well. It's been a
Oh
while.
my
Like,
gosh,
look,
the obligatory unnecessary pleasantries.
yes. And then
Mhm.
I have the worst, but
Yeah.
you are a scientist like myself. You have a PhD.
Mhm.
And like you said, you're getting back into the cannabis space. You're a plant person. You teach classes and
Thank
you do
you.
stuff that I have no idea how to do. And I'm learning a lot from you on your TikToks when you talk about
Okay,
tissue
awesome.
culture and stuff like that. And
Okay.
these are definitely skills that I need to have or at least know about working with my flora. And so, obviously, you know you better than I know you. So, go ahead and tell the folks who you are and what you do and why you're in the cannabis space a little more.
Yeah, Chad Ternes, originally from Oklahoma. I did all my schooling there. I went to OSU, Oklahoma State University, bachelor's in botany. I did switch majors about 6 times before I landed on botany. It kinda hit this point of, yeah, started dabbling in cannabis more. I had been consuming it for a little while at that point, but I kind of decided that I really love plants and we were growing it at the time. We were growing it. I loved it. We did just really simple deep water culture, hydro. And I was like, man, I want to do this. And my initial thought was, I want to do metabolic engineering in cannabis. And that was back like late 2000s. So
Mm.
2005, six, somewhere in there. But nobody was really doing cannabis. And so it's kind of, well, I'll just adjacently work my way. And did grad school at OSU, and that it turned into a PhD unexpectedly. And that was actually on algae.
You're
But
like, I accidentally did a PhD.
well, my wife and I, we decided to get married, and she decided she wanted to do a bachelor's. And so she did that. And I was like, well, if we're going to be here like 4 more years, then I'll just do a doctorate because my advisor had been trying to talk me into switching from a master's to a doctorate up until that point, anyways. So I
That's
was
how they
like,
get
okay.
you.
So in hindsight, I probably would have done something a little different, but yeah, grad school at Oregon State fell through last minute doing a master's there. So kind of OSU, Oklahoma State was my backup and did that. And the algae was cool because that's really where I got into. That was all bioinformatics for the most part. So lots of genome annotation and analysis. And a big part of my role in that was just annotating metabolic pathways in this algae. It's like a thermoacidophilic red alga. So it lives next to sulfur springs, volcanic calderas, and stuff like that. So interest for like biotech purposes, right? Like
Mm.
thermostable enzymes, stuff like that. And then, yeah, got married, had a kid, and decided to do a postdoc. So went to Michigan State, was there for a little bit. That was personally a disaster. In the lab, I kind of typical avoidant ADHD, just head down and just work, and did fine in that department, but it just didn't really work for the family. So back to Oklahoma, I did another postdoc at Oklahoma State, and then eventually made my way out to Colorado. So here at Colorado State, and that's been off and on, but it's all centered around just a lot of molecular work. And so ultimately, I wanted to get into kind of bioengineering, synthetic biology. And again, I wanted to do that in cannabis from the very beginning. But as time has gone on, it's kind of that's shifted a bit, to be honest. I still love cannabis and will probably always work with it in some way, shape, or form. But I guess my interests and my kind of the direction is changing up a little bit. It's kind of rather recent, honestly. Yeah, I love science. Like I geek out on plants. I really do. Uh, and especially molecular stuff is
Yeah.
really kind of where that is. I really want to get into this idea of like biodesign. And there's people doing it, several actually, but really kind of this intersection of synthetic biology and like design, art, architecture, and stuff like that. Because as a kid, I wanted to be an architect. Like that was my dream. And I got to college, and for whatever reason, I convinced myself that I wasn't creative enough to be successful as an architect. And so I just ended up not doing it. Literally talked myself out of it with a thought. So stupid. Don't
There's
believe
still
your
time.
thoughts.
You're still young. You could do it.
Oh, and that's the thing. It's funny you say that. So a few years ago, I saw this episode on Netflix. They have a docu-series called Abstract, The Art of Design.
Mhm.
And in season two, there's an episode about Neri Oxman, and she is like a classically trained architect, but got into biotech, synthetic biology, and all of that. And she was a professor at the MIT Media Lab. And but anyways, she's like, yeah, this merging of biology and architecture and all of that. But when I saw that, I was just like, man, that's what I want to do. And then
Mm.
again, I was like, but that's like Boston or the Bay Area, and I have kids, and just the avalanche of stupid thoughts of why I can't do this. And really, more recently, kind of in the past several months, really, it's kind of coming back to that of why can't I do this? And I think that taking a quote unquote orthodox route of academia and stuff like that, I don't think that's 100% necessary. I used to think it was, but now I'm not so sure, because at the end of the day, I can build a lab pretty much anywhere. Does it cost money? Sure. But we're scientists, we're hackers. And so, kind of, yeah, pivoting a little bit to do more, yeah, exploring this intersection of like biology, specifically plants, art, design. And I think really cool within that is if we look at just the natural plasticity morphologically within plants, right? We cannot alter a plant's genome in any way, shape, or form. And the different morphological features we can get out of it, right? Like training it or whatever. It's really kind of remarkable the capability that's already within the genome natively, right? With just a little tweak here, a little tweak there, whatever, that just happens sheerly by chance because it's nature. That's really what's been kind of piquing my interest lately in trying to probe that. So even now, looking at, okay, what is a kind of a model plant? And I really kind of curious about ornamental plants because I think that field is prime for some really cool stuff. What's kind of like a model plant I can start playing with to really start kind of probing these questions, if you will. And that's going to be, yeah, a lot of tissue culture, which I'm doing that in the lab for my kind of day job, if you will, at the moment. And it's kind of funny how that has bled into like, oh my gosh, this opened up this whole new avenue that I kind of just thought was dead for me, so to speak.
Mm.
So yeah, getting really excited about that stuff again. And finally getting to work on my problems that I want to answer science wise. I think that's one of the downsides of doing postdocs and stuff like that is like until you're a professor with your own program or whatever, you're kind of doing everyone else's research for the most part. Like, yeah, it's yours, it's your name on the paper, assuming it gets published, but it's really kind of, even now, it still doesn't feel like my work necessarily, even though what I'm currently working on, I've like designed all of it and cloned it all and did all of it from kind of beginning to end, so to speak. It still doesn't quite feel like mine, even. And there's not like an emphasis on mine, but it's more of, it's not the direction I want to go in, right?
Yeah.
It's
Yeah.
more, yeah, it's still adjacent, but it's kind of minor tweaks, if you will, to start to kind of do the research that I want to do.
Yeah.
So,
I can appreciate that. I never did a postdoc. Well, the PhD thing, like I never thought I would do a PhD. I never really thought I'd do a master's. Like, so
mhm.
I did 5 years at CSU and my major was anatomy and physiology and I thought I wanted to be a vet and I turned 21 in my last semester of my 5th year, and I got a zero point 0.0 GPA on 18 credits. My parents were not happy. I just I was just partying, and I was partying so hard that I just forgot to like withdraw from my classes or
Mhm.
whatever, or go to class or even do anything. I thought, well, okay, I'll just work because I've got 12 credits left to graduate. I'll just work, get the money, and then finish. But in that summer, well, in that little break that I was taking and working, I accidentally got pregnant and then had a baby. And so then
Mhm.
I worked in pizza. And then eventually I was like, I kind of want to go back to school. So I went back to school and finished. And I had to transfer because I had moved to Greeley and was running a Papa John's in Greeley then. And so I was like, well, I'll just transfer my credits over to UNC and finish. They don't
Mhm.
have anatomy and physiology as a major. So I switched to
Okay.
cellular and molecular biology thinking, I could go into cancer research or something like that where the money is.
Mhm.
And I had to like redo some classes, including like organismal biology, because I couldn't pass organismal biology because of the stupid plant part, right?
Funny how that happens.
I It was just, like, so foreign to me, I didn't get it, I don't understand plants, I don't like plants, why do we have to learn about stupid plants?
Right.
Nobody likes plants. And then the other thing, I had to take an upper level division biology class, and I really wanted to take animal behavior, but it was full, and there was a wait list, and there was no way in hell I was getting into that class, and the only other class that was available to take and that would allow me to graduate on my timeline was a stupid plant systematics class, another plant
Okay.
class. And I was
Right.
like, "All right, I'll take it."
Classification at that.
Oh, so boring. But it was a brand new teacher, this young guy, he was, like, super energetic, like, really enthusiastic about plants, and he had the plant population genetics lab. So the class was fun, but, like, okay, so in addition to the 100 must-know plants on his list, right? We had to learn those. We had to go and collect out in the field 20 additional plants. And I was like, "How? There's 100 on the list. That's all of the plants in Colorado. How are we gonna find
Right.
another 20?" Like, that's how much I didn't know about plants.
Mhm.
So, and then we had to collect the plants. I barely found 20, but I found 20. We had to make herbarium labels, and we also had to sketch the plant. And the idea behind that was like actually look at the structure and function and
Mhm.
all of the bits and parts and of the plant and how different they all are. So it was a good exercise in that. And then, all right, so the class wasn't too bad, and I don't know, I probably got like a B in it. And it was Mitt's first semester teaching at UNC, and his tests were really hard. But I had to also do a capstone credit to graduate on time. I had 6
Mhm.
capstone credits, and I had not done any. And it was my
Okay.
last semester, and I had one class, which was molecular genetics, and then and nothing else, and that was just on Thursdays. It was like a 5-hour class on Thursday because we had to do, like, lecture and lab, like, at the same time because we ran PCR and stuff like that. And yeah, so I was like, "Oh my god, I how am I gonna get 6 credits in?" And so I approached Mitt and I was like, "Do you have anything in your lab that I can do?" He's like, "Oh, we're working on this cactus project with the BLM and the Denver Botanic Gardens. 6 credits is a lot. That's like 100 hours." He's like, "That means you'll be in the lab like every day." I was like, "I got time. I don't have any classes like this." And so, yeah, so I started this project with these cactus, learned DNA extraction, running PCR and gels and stuff like that. And I kinda liked it, and while I was working in his lab, Jerry, his PhD student, and I would, like, chat, and I was like, "I think I wanna do a master's." And so I asked Mitt, and Mitt was like, "No, I don't think this is for you." And he didn't want me to do it, and then
Okay.
he's like, "I don't think you'll like it." And I was like, "I will honestly like anything I do, and I think I wanna do it." And it's like, "Alright, well, keep going with this project that you've been working on, this hybridization project." And so, yeah, that's what I did for my master's, and then I worked at the Denver Botanic Gardens for 2 years, and that's when Colorado legalized adult use, and I was like, "There's some pretty easy to answer questions with my population genetics background, like
Mhm.
hybridization of really closely related species. Like we've got one
Right.
species here, or maybe two, or maybe three, depending on what you subscribe to. But really, the question that I had was how come when you go to one dispensary and you buy Blue Dream, and then you go to another one and you buy Blue Dream, they're not the same
Right.
because they should
Right.
be. And I was like, that's a super duper easy question to answer. So I approached him and I was like, can I do a PhD? And he's like, no, absolutely not. He's like,
No.
he's like, "This is a conservation, genetics, and rare plant lab. We're not doing weed." And I was like, "Oh,
Mm,
no." So I walked out of his office with my tail between my legs, and then he sent me an email, and I thought he's like, "Can you come into my office?" And I thought it was something to do with the sclerocactus project. So I came in, and he's like, "Do you still wanna do this weed project?" And I was like, "Yeah." And he's like, "All right, you're in. I already talked to the lawyer and the provost and the dean and the president and the chair, and they all said, you know, the project sounds good and it's fine. We can do cannabis." He's like, "But I don't know anything about it. I don't know where to get money. You're on your own. I'll here. I'm here to rein you in and like keep you in the rails, but
yeah.
like this is gonna be all you. I'm not gonna be the weed guy. You're gonna be the weed girl." And I was like, "Okay, let's do
All right.
this."
Fair enough.
So like, my PhD is kind of a accidentally did it kind of thing because I had questions and it was like the best way to do it. And it was just a
Mhm.
perfect storm of having Mitt being as cool as Mitt is, the
Yeah,
school being as
that
cool as
helps.
the school being as cool as it is. You know, they
Mhm.
had a cannabis program in just about every department, like the exercise and sports science, recreation and tourism,
Mm,
the business school, the chemistry department, like you name it, they were doing
yeah.
something with cannabis. And so yeah, it was just like a perfect storm to be able to do it, and people still question, "Where the hell did you get a PhD in cannabis from? There's no schools out there that do that." And I'm like, "There still isn't." You
Right.
know, it's either hemp or, you know, non-plant
Mm,
touching kind of stuff. So,
mhm.
but
That's certainly, yeah, the kind of the perfect storm of legalization here in Colorado, right? And just access to all different samples from various
mhm.
places. So that's, yeah, that's cool that that worked out like that.
And I did have to take out student loans, and I know a lot of people buy pot with their student loans, but,
Mhm.
like, I didn't buy it to smoke it, I was buying it to do research
There
on.
you
Yeah. So I did an accidental PhD too, but I've never done a postdoc because I was like, "Well, I'm not following the traditional academic route, right?"
mhm.
I never wanted to. And, you know, I worked at the Denver Botanic Gardens, I ran their conservation lab for a couple of years, and I'm like, "Why do I need to" I still don't know why people do postdocs. Is it just like a transition into getting a real job, or is it like you just not ready for the real world because 'cause you're weird, or
I don't, for me, it was since most of my PhD was computational work. I wanted to do more lab work. I had done lab work. We did a lot of cell-free protein expression. So my advisor did. He was an ion transport guy, so like biophysicist, basically. So I did some of that, but I wanted, yeah, more molecular stuff, like hands-on bench work. And he honestly kind of tried to dissuade me from doing that completely. He was like, I don't know. He was a little ambiguous now when I really look back on stuff. Like well intended, but I don't know. He had never been outside of academia. He was, yeah, from Germany and
Mhm.
all of that. So, and I probably made the mistake of not asking more people for just kind of advice, wisdom, whatever. But I had this idea that, yeah, I got to do a postdoc to get this experience. And I guess, yeah, I go to industry, but I really never intended on staying in academia for as much as I have, for as long as I have. That has, I don't know, it's kind of the door that just kept opening up for me when I needed a job, for example. And I think what you mentioned is kind of you didn't want to do the traditional kind of academia route and all
Mm,
of that. And I think for myself, I really enjoy teaching, but I don't necessarily want to do it at a university, or at least not a traditional university. I want to do way more hands-on, like in the lab intensive type stuff. I actually really love teaching labs in grad school. And even now I enjoy working with undergrads and stuff. So I don't think I ever necessarily had a clear direction. It's kind of taken me a long time to get here, if you will, to kind of have more of an idea of how I want to move forward with science and things of that nature. But yeah, traditional academia is it's kind of brutal, right? There's not a lot of positions open. There's tons of people applying for positions. I know when CSU had an opening here in the bio department, there were, I mean, over 100 applicants, for one spot. And I think well over 100. And that's tough. So tenure track positions are becoming harder and harder to get and fewer and further between. And what we're seeing is an increase in like adjunct positions, so non-tenure track type stuff.
Mhm.
And I don't know. It kind of just depends on what you're ultimately after. If you just want to teach and not run a research lab, go for it. Teaching and research, like typically something takes a hit on some
Mhm.
level, unless you have a lab that's just people around everywhere and you have the funding to it's
Keep
kind of
going.
a well-oiled machine, if you will,
Yeah.
but not everyone has that luxury. So, and I think, yeah, you're starting to see more cannabis in academia as well. Here at
Mhm.
CSU, there's I think at least a few different classes I can think of that are now specifically cannabis or hemp. Well,
Mhm.
it's the same damn thing. But that's another discussion. Hemp versus marijuana versus cannabis. Like it's all cannabis, damn it. So
It's funny that like, well, it's not funny that you enjoy teaching. It's awesome that you enjoy teaching, and you teach stupid plant classes, right?
yeah, so it's yeah, synthetic biology is what I'm teaching right now. And then for the past year online, I've taught medical botany. So that's been
Okay.
fun actually. So like kind of ethnobotany, ethnomycology, that type of stuff. That's
That's
been
interesting.
fun.
Yeah, for sure. So it's
Hmm.
it's it's funny though, because like UNC, all of their PhD degrees are in education. So if you're in the physics department, your degree will be in physics education, chemistry
Okay,
education, biological education. Because
gotcha.
when they wanted to build a PhD program, the state was like, "Well, we already have two schools that have PhD programs. Why do we need another one? And what would set your school apart?" And so they were like, "Well, we're going to be the education school." And
Okay.
so my degree is in biological education, but my research focus was on cannabis. And so really, and I didn't even know that when I started the PhD program. Like I found that out like probably 2 years in. And I was like, "What?" I was like,
Hmm.
"'Cause why do we have to take all these teaching classes, like learning how to teach and teaching how to learn?" And we had to take, you know, this statistics class where they talked about how you were gonna measure your students' success and how would, and I'm like,
Hmm.
none of these statistics like can be used for biology. Like, why isn't there a statistics class for PhDs in biology? Oh, because this is an education school. Like everybody's
Hmm.
here to do a degree in education. I was like, I'm not. I don't want to be a teacher. I'm
Right.
not interested in that at all. Turns out, well, I ended up flipping the anatomy lab to a flipped style classroom. So
Hmm.
the students, and it happened because there was a giant snowstorm and we knew that classes were going to be canceled on Thursday because of this storm and possibly
Okay.
Friday. No labs on Friday, but everybody that took labs on Thursday was going to miss a lab. And that's a whole week of lab work. That's a lot to miss
Yeah, it
for
is.
anatomy and physiology. And how are those students going to make that work up? Like it's just impossible. That's 6 labs times 30 students. So
Mhm.
my friend Vanessa and I were like, let's videotape us walking the students through every single word, showing them on the model, and then we'll put it on YouTube and then they can learn it. And so we did that. And then we
Awesome.
were like, let's do that for every single, all the words that they have to learn. And we
Mhm.
did that. And then we were like, what about if we make the students watch those videos before they come to class? So that by the
Mhm.
time they get to class, they've already seen it once. They take a quiz on it when they get to class so we know that they did it. Then
Mhm.
when they're in class, they can ask questions of things that they don't understand, or we can, help them out a little bit more. And then they can they have to label their models and everything. So label the models. Then they take pictures of that. They make a PowerPoint. That's their homework. They send it to us. We grade it, but it's also their study tool. And so we totally flipped the classroom to where they're doing most of their learning outside the class and they're coming to class to study. And
Mhm.
we raised the traditionally students don't do well in A&P classes because they're really, really hard. It's really hard to
Yeah.
do all of that rote memorization
Mhm.
in addition to like the lecture material, which is, more the physiology part of the class.
Yeah.
And so the average grade in physiology, anatomy and physiology labs was around C minus.
Okay.
Um, we brought the class average up to a B plus doing
Wow.
it that
Yeah,
way.
that's impressive.
Students had a lot more fun because it's
Hmm.
that class is so stressful.
Yeah,
And these are the
a
students
lot
that are
of.
going into pre-med and nursing, and if they can't pass anatomy, they don't get into the program. But I never wanted to be a teacher. I never wanted to publish on education, but we published 3 papers out of that. The first one was called, um, It All Started Because of a Snow Day. And
Mhm,
then the next one was, I think, called Yes, Use Your Cell Phones. So
mhm.
they use their cell phones to take the pictures. And we figured if they're gonna, I don't like teachers that are like, put your phones away, don't look at your phone. I'm like, make them use it. They're gonna have it out anyway. Like if they
Yeah,
have to use it during class, they're not gonna be on Facebook updating their status.
right.
So so making them use their cell phones and teaching them how to use Google and PowerPoint and things like that. It's
Mhm,
amazing how many students don't know how to use Google. They know how to use social media, but Google is just like
mhm. Just basic computer literacy outside of social media or whatever. Yeah, that's the thing. Teaching students Excel. I remember Plant Phys Lab, just how do we put data in Excel and make a graph from it, right? Just
Mhm.
some super simple stuff, but yeah,
So I never
the
wanted to be a teacher. I never wanted to work with plants, and yet here I am.
mhm. Like, yeah, I have kind of an adjacent story. So one of the guys on my grad committee, Dave Meinke, he was like kind of considered like one of the godfathers of Arabidopsis genetics, if you will. And he told me the story about when he was in grad school at Yale getting his doctorate. One of the guys on his committee, or another professor, I can't remember, he was a developmental geneticist. And he kind of, I guess, low key insulted Dr. Meinke back when, of like, you're hardly even Yale material, made a comment kind of like that. And Dr. Meinke, Dave, took that personally, I guess. And so that's actually what he ended up like doing his whole career in science over, is developmental plant genetics.
In spite of somebody making
Kind
a
of
comment.
kind of seems like that, really. And what's funny is he was on my grad committee, and I never got insulted like that, thankfully, at least not by him. And kind of lo and behold, through all of this, it's something that I've been becoming more and more interested in, is developmental genetics. And actually in my orals, my oral comps, that is what he grilled me on, was plant developmental genetics and like anatomy morphology. And I was just like, when the hell am I ever going to need to know how many cells make up a female ovule in a plant? It's like 7, by the way. And then, yeah, on my comps, my advisor made the comment of, oh, we should close the window in case Chad tries to jump out of it during this. Grad school is not for the faint of heart. I mean, I think there's a reason it has such a low completion rate for a doctorate. And
Yeah.
so, because like I almost quit. I was so close. I was 3 years in, I think, and I was not doing the work I wanted to do. I started doing a lot of phylogenetics. I started doing a lot of like phylogenetics, like evolutionary history type stuff. And I was just kind of miserable in it. It was like, man, why am I doing this? And I ended up just kind of grinding it out and finishing. I was like, well, I've already spent this much time and money, and to quit now and not have anything to show for it. And that was kind of literally what got me through, was that.
I loved doing my PhD. I loved all the work I did. I loved all the questions I was answering. I had a blast. I had a rule. I was only going to work 9 to 5. We'd never study past 10. Weekends were not no school. So I really was really strict about that. But I loved it. And I did it in 3.5 years, which is like, what?
Okay,
What?
nice. Yeah. Wow, that's insane.
And I still have an unfinished project sitting in the lab on it's an epigenetics project, but it was like, you're done. You have to write your dissertation and you have to graduate. Because I had already
Mhm.
done 5 whole projects, 5 whole experiments or whatever you want to call it. And he's like, that's enough. That's
Yeah,
enough.
that's plenty.
He's like,
Plenty.
"You're gonna have 7 chapters you have to write and you're done." So he cut me off, and that was the whole purpose that Mitt was there for. It was like to reel me in and not let me keep going because he knows that I wouldn't stop.
Mhm.
But no, I love doing my PhD. But the oral comps were rough. I did enjoy the final dissertation, like your defense of your PhD, that freaks a lot of people out. For
Hmm.
me, I was like, this is the icing on the cake. This is my chance to tell everybody what the hell I've been doing for the last 3.5
Right.
years. Like, I can't wait to like tell everybody. And people are
Hmm.
like, you're so fucking weird, Anna.
No, that's an awesome mindset to have about it, though. Could
Yeah.
see how that would be way more beneficial than fear of just everyone's going to ask me every question that I don't know the answer to.
And they did. Like, I got some really so at UNC, we do it's open to the public. So
Hmm.
everybody comes in, then they excuse the students and the general public, and it's just the faculty. They ask you all kinds of questions. And then they all leave, and then it's just your committee that's left, and then
Hmm.
they ask you more questions. That whole thing, me defending my thesis, was like 3.5 hours, like a ridiculous amount of time.
Hmm.
But I got really good questions and I was able to answer them all for the most part. Somebody asked me about terpenes and like what kinds of insect or is it for defense or what? And I was like, you know what? I can't answer that question, but what I can say is that we've been heavily selecting this plant for traits that we enjoy.
Hmm.
So no, those are not necessarily the things that the plant needs to survive and be able to have good defenses for various pests and pathogen pressures, because that's not what the plant is doing. It's surviving because made it that way.
Right. We like smoking the hell out of it. So,
Yeah. So, and thinking back, looking at cannabis now, we've been selecting for higher and higher THC and louder terpene profiles, but we're
mhm.
seeing more diseases. And
Yeah.
because the plant is not able to, like, it hasn't been naturally adapting to things that it encounters in the wild.
Right.
This is
Controlled
completely,
environment.
yeah, like there's no such thing as wild broccoli. Broccoli is
Hmm.
a fully man-made thing. We selected for it to be like that. You would
Yes.
never find wild broccoli growing, and you really wouldn't see the things that we grow in our cultivation. Like those are not
Yeah.
natural.
It's like almost everything we eat does not look like its ancestor, right? Corn, teosinte, all of it. Tomato, nightshade ancestors. Just it's yeah. The
Strawberries,
selective breeding that has
bananas.
happened is insane.
Hmm.
Right. For sure. And it's yeah, that's kind of touches on a few different things, really, of like kind of this we're babying all the plants, controlled environment and stuff like that. And I think Dan Fowler has raised this point pretty heavily as well of the plants are just kind of total pussies
They
when
are.
it comes to stuff.
They're big
And,
old babies. They
you
don't know how to
know,
do anything.
and I think you, like you said, we're chasing 35, 40% THC, if that's even possible, loudest terps and all of that. In my experience, it's like the stuff I enjoy smoking the best. It's hardly ever 30% THC. Usually it's like maybe 20 or less, high teens even.
My purchasing decision that I walk into a dispensary and I'll ask, "What's your lowest THC that you have
Mhm.
on the shelf?" That's what I want.
Yeah. It's like some of it's just, I don't even enjoy it because it's
No.
almost not that it's too strong, but it's like you're missing all of the different nuances that this plant has, right? Like, I think that's one thing that really drew me to cannabis and like just metabolism initially was it's like an actual just chemical symphony, if you will, within
Yeah.
cannabis. Like, it's the amount of compounds it can make is absolutely insane.
Right.
And we keep discovering more all the time. Like,
We're stripping it away by
we are.
selecting for single traits, and the complexity and the layers of things that would contribute to the synergistic effects that you get are just being lost.
Hmm.
And so it's no wonder that, and I feel really bad for people who are like, I think I'd like to try cannabis. And then they go to the store and they get what they think is a quality product because it's on the high, it's on the high shelf. It's the platinum product, whatever. It's got high THC, which everybody thinks is how you gauge a quality product. So they take it home and then they just don't fucking enjoy it. And it's
Right.
like, yeah, no, you probably won't. It'll probably give you a panic attack, like,
Hmm.
and so I think there's probably a lot of people out there that are like, "Oh yeah, I don't like cannabis." And it's like, you might if you didn't have such a slim selection of basically shitty weed.
Yeah, essentially. Yeah. And I mean, I remember, so I went several years without smoking. So when we first moved to Colorado, like I didn't smoke at all. And for the first few years, I didn't smoke while we lived here. And my wife at the time was just like, you should start smoking weed again so you can chill out a little bit more. And because, yeah, I was way more laid
Sure.
back. So a little, well, yeah, a little like kind of anxiety, stuff like that, and just kind of a nervous wreck in a general sense. But so what I started doing, started back smoking with, if you will, was a friend's parents had come to visit from Hawaii. That's where they were from. And her mom got a pen while she was in town, just like a disposable pen, distillate, and left that because she wasn't going to fly back home with it. And so I started hitting on that. And I think the first time I did it, I took like 2 hits off that pen and I was just freaking gone. I remember we were watching Queen's Gambit on Netflix, and I remember laying on the bed and I was just like, yeah, I don't think I should have hit that twice. So I can see how if somebody is not used to it or they're like brand new to cannabis, have never done it before, if they're kind of steered towards an incorrect product for their experience level, if you will, yeah, I could see how that would absolutely freak people out. And that's not going to be a good experience for anybody. And
Right.
so I do think, even you say, like, let me see your lowest percentage THC, it's like, I don't even know if there's really like that big of a spectrum when I walk into a dispo. It's kind of almost everything is 20 plus. You might have like
Yeah.
the one, maybe two options, typically in the popcorn range, kind of lower tier stuff that's middle high teens or whatever. So,
Mhm.
but yeah, some of my favorite strains that I've had in the past few years, it's like 17%,
Mhm.
18%. I'm like, dude, that stuff slaps. So
Is that what they printed on the bottle, or is that what you tested it at? Because
that's what's printed on the label. I haven't done any potency testing myself. My lab I work in isn't really made for chemical analysis like that. Like we don't have a GCMS or anything like that. Have access to it, but yeah.
I was gonna say we grew some in Colorado that we had tested, and my we forgot to label it because I'm a terrible scientist. Gavin called it "dick in the dirt" because it just would knock you on your ass. But we had it tested, and it tested at like I think it was like 12%.
Wow.
Yeah,
Dang.
and, I did that whole study on potency inflation, and basically what I found is that most of the numbers are inflated an average of 30%. So if you take
Yeah.
your 18% weed and you multiply it by point 0.7, so 70% of that, then that's the weed that you said slaps. That's
Hmm.
about 12%.
Yeah.
And the last one that I bought was called cherry water ice, and I think it was 18%, so right around 12. And
Okay.
so if anyone's watching, like, I feel like that 18% is kind of a sweet spot.
We have some stuff right now called cereal box that it's a hybrid. We really like it.
Yeah.
And it's like, yeah, 17, 18%. It's right there too. I was kind of surprised how much I liked it,
Mhm.
but it smells awesome, kind of like Fruity Pebbles a little bit. It's good. I enjoy it. I don't know what its like parents or lineage is. I asked them, but they didn't remember offhand.
Could it be cereal milk and something maybe?
That would be my guess. But I'm not, the next time I go back, because I'll have to go back in a couple more days, I'll ask them again and see if they remember, because they grow everything on site.
We're not allowed to grow in New Jersey, so that sucks. And, I don't smoke a lot, but I do enjoy growing it.
Mhm. Growing, it's fun.
And New Jersey's stupid because I think they think that if people can grow it at home, then they won't buy anything from the dispensaries, which is dumb
Yeah.
because you can only grow so many plants. There are a lot
Mhm.
of people that can't grow because they suck at it or because they don't have the
Mhm.
space. And
Right.
also, when you grow your own, like you don't want to smoke the same five. all year round. Like, you
Right.
want some variety. They have homegrown in tons of other states, and the dispensaries do just fine, you know?
Yeah. That's like saying you can't grow tomato plants because, right, nobody will buy them from the grocery store. It's like,
Right.
it's just asinine, frankly, because you're gonna have people that just like you said, they can't, space constraints, whatever it may be. People who just don't want to put the time in to do it. They'd rather just go to the store and buy it and be done with it. Like that market's always going to be there,
Some
I would
people are
argue.
just not interested in gardening at all. Yeah. So I have to ask you, what does a synthetic biologist do? What is it? What is synthetic biology?
Yeah. If you would ask some people, they'd be like, well, it's just bioengineering. And that's a component of it. But really, the idea behind synthetic biology is we want to be able to basically program life like we program computers, right? So if we can kind of quantitatively characterize a gene, so output or like whether it's promoter strength or RNA half-life, terminator, whatever, if we can quantitatively characterize that, we can then use like mathematical modeling to essentially build genetic circuits, right? This kind of just goes back to basic logic gates, essentially. So strip everything down to binary, so on or off for the most part. But yeah, kind of treating genes and quantitatively characterizing everything so we can then use modeling to precisely tune, if you will, a particular response or production of a metabolite or whatever. In my case, in a plant. Yeah, I don't mess with animals. I just like plants. Plants don't scream or, yeah,
Oh, they
bite.
do. The smell of fresh cut grass. That's the smell of grass
That's
screaming.
true. Oh man, but that's the greatest smell though, right? That's like probably my favorite Hank Hill line is why would anyone do drugs when they can just mow a lawn?
Do you
So,
think that, like, there's a whole bunch of people out there that think there's a bunch of genetically modified weed. And now, depending on how you think about genetic modification, I'm in the old
right.
school camp where I think that if humans have altered the evolutionary trajectory of an organism, it is genetically modified. So
I
therefore,
agree.
all cannabis is genetically modified because there's no natural cannabis anymore. I think what people are thinking is genetically engineered. Like
Mhm.
somebody's gone in there and inserted a gene or taken out a gene or
Right.
knocked out a gene or something I guess you could call polyploids genetically
Mhm.
modified as well. But if we're talking about like specifically fucking with the genome, inserting or taking out a gene intentionally, the only one I've ever heard of is Daniel Fowler, and who is it, Canopy, that has the patent on that. And then there was another group a few years ago who was able to genetically modify a cannabis plant to produce water soluble cannabinoids, which I was like, whoa, that's crazy. But it hasn't come to market and I never heard anything about it. So I feel like some of this genetic modification, some of this more intense genetic modification in cannabis, while it works okay in the lab, whether or not it works at scale is another story. And I feel like you're probably better suited to talk about that than I am. And I know that there's like tons of repetitive elements in the cannabis genome, which I feel
Mhm.
like can make it very problematic to try and get, a genetic modification to do what you want it to do and not fuck up something else unintentionally.
Mhm. Well, yeah. So for as far as genetically modifying plants, like actually inserting a foreign or transgene, putting it into a specific place in the genome is not trivial at all. And that's actually pretty difficult. And pretty much the approach that almost everyone takes. That's how we do it. It's just random insertion. We don't know where it's actually getting inserted into the genome. Right now, we use agrobacterium to do our transformation. Typically, agrobacterium or biolistics are going to be your two main routes. Biolistics being literally just a gene gun. So you would coat gold nanoparticles with DNA and then literally shoot them onto a petri dish of cells and just sheer blunt force of blasting through the cell wall, literally using a gun. Imagine a shotgun, and that's biolistics for the most part. Agro is a little more, I don't want to say elegant, but I really like agro because it's really simple. You're only going to get one insertion into the genome. When you're doing biolistics, you could actually have multiple insertions in the genome, and you have to follow up testing that molecularly, what your actual copy number is. Agro is just nicer, it's cleaner, it's
Like more specific or more intentional, maybe?
yeah, well, like agro was it's a plant pathogen. Like it literally evolved to infect plants and infect the genome. And so over the years, decades now, we've kind of hijacked it a little bit to kind of get it to do what we want it to do for the most part, as far as DNA, plasmids, and stuff are concerned. So that's kind of the two approaches.
Mhm.
As far as is there genetically modified stuff out there, meaning we've inserted a foreign gene into the genome and that strain is now out in the public, highly, highly doubtful. I know way back when I would hear about like G13
Right.
was right genetically modified, and I was like, maybe, but again, like, how'd you modify it, right? Like, what did you give it? Antibiotic resistance? Did you give it a pigment biosynthesis gene to make a new style of pigment? What'd you actually do with it? Did you just have a CAM35S and promoter, and that's just driving whatever? Like, what the hell did you do? CAM is derived from a viral, from mosaic virus, cauliflower mosaic virus. And it's just, we just call 35S, but it's just a highly constitutive promoter, meaning it's just always on and it's always expressing at a high level. And I don't even know how well that promoter would work in cannabis. I don't know if there's any papers on that. But yeah, there's probably not much, if anything, out there that's actually genetically modified in the sense of engineered it in a lab with putting genes into it. Gene edited, that is that's kind of an interesting distinction because if you call something a big distinction is you can use genetic modification to create gene edits. CRISPR is this tool we use, and basically it can do two things. It can locate a specific place in the genome, and then it can cut the DNA at that site, double strand break. Okay. And typically, when that repair mechanism happens, the cell's own repair mechanism for fixing the DNA, it'll often make mistakes when it makes repairs. And oftentimes, when those repairs are made, those mistakes get incorporated, that can deactivate the gene, right? We messed up the coding sequence, the translation frame, whatever it may be. So, a lot of time we have deactivated genes as a result of CRISPR gene edits. We can do specific template repair, but that gets a little more tricky. And so you can have a plant that, okay, we modified this plant, we put CRISPR into the plant. This plant is now genetically modified. And then the CRISPR machinery in that plant made the gene edit. So it cut it, fixed it. There's a mutation, it's edited. And that may just be less than 5 base pair change, right? Plus or minus one, really. And but you now have your plant that has the edit, still has CRISPR. We can breed that with other progeny to breed out the CRISPR machinery, but
Mhm.
keep the gene edit. So now you have a plant that is gene edited, but not genetically modified. That's kind of like, I guess, the nitty gritty detail kind of definition, if you want to get at. And even the USDA years ago came out and said, yeah, gene edited is not considered genetically modified. And so I'm pretty sure you had some companies come into existence when that announcement was made. And I think a big one in the Boston area was one of those. So,
Oh, okay.
yeah, follow.
Yeah.
But so that's kind of the gist of that. As far as doing cannabis, it's kind of historically known as a pretty recalcitrant species, meaning it's really hard to genetically transform. And it's just kind of how it's been. And, we can kind of take stuff down to this like callus tissue level, transform that, or like even the protoplast level, which would be plant cells without cell walls, and transform those. But then you run into the problem of plant regeneration. So regenerating an entire plant from a single cell yes, it can be done, but it's not trivial. It's not as simple as some people think. So there are definitely some kind of roadblocks, if you will, and regeneration was a big one. I think some people are have found ways around that. But yeah, actual gene editing, modification, engineering, if you will, of cannabis using a kind of lab molecular route, I don't think that's nearly as common as some people are led to believe. So I would highly call bullshit on a lot of it, honestly.
Yeah, I would agree because CRISPR has been around for quite a while now. Yeah, and if it was successful, where's all the CRISPR modified plants at? Like
Right.
cannabis, it's just not they're not around. And so like while I applaud people who are interested in using CRISPR to do whatever in cannabis, I'm like I don't see it as being any more effective, cheaper, or faster than our regular, breeding, traditional breeding. Honestly, there's some really talented breeders and growers out there that are doing a great job of creating new things and pulling out traits that are different.
Mhm.
And so I'm like, why would you do it in a lab when we can do it in a greenhouse?
Yeah. Well, it really, I think it gets at like, what is the specific genetic mutation that you're wanting to change?
Mhm.
And honestly, when it comes to like model plant species in science, like Arabidopsis is the one, we have gene knockouts for like so many genes, like it's not even funny. And so we know if we knock this gene out, we know what result we're going to get. We don't have that in cannabis. It's like, yeah, we can knock out like in Dan Fowler's the canopy gene editing thing. I think they knocked out like phytoene desaturase, right? So you get an albino mutant. That's a very easy way to see, oh, we have our plant because it's white, it's not green, right? But when you're talking about trying to engineer, edit, whatever, a cannabis plant for commercial production, terpenes, whatever, what exactly are you going for? Are you just going to knock out this one gene that's upstream from this product that you don't want to make, like a terpene that you don't like, and we want to make more of a different terpene? What's your exact approach for that? And so, using kind of molecular tools, the molecular engineering route, if you will, we can kind of be a little more specific. And when we're doing the traditional breeding route, I think is very powerful and will always be relevant. We may not have a representative plant that has the trait we want, right? Because if we have, say, we want this trait, but we don't have a plant that exists that has this trait, well, then how are you going to make that trait? You can't breed towards it if it doesn't exist. So
Right.
that would be kind of one instance where the lab approach would be superior to just traditional breeding. But I agree, like really the true breeders out there, the people that are growing out thousands and thousands of plants to really see not only the variation, but I mean, like you're a population geneticist, like that's really where you start finding that cool stuff. And it's like, I guess, along the lines of a pheno hunt, but don't just stop at hunting your pheno and just that's the one, right? This idea of stabilizing the genetics, if you will, because if you're just like breeding two parents together, your F1, you're going to see insane variation with if you grow out enough plants. And it's like, what traits are stable, what are unstable. And that's really kind of, I think, where you have like the Phylos of the world and people like that, or I think probably Trilogene as well. It's like growing from seed instead of clones, but having the genetics have been kind of stabilized over enough generations to where you're going to get that uniformity across a population.
And it's
So,
tough because, cannabis breeders are not afforded the same protections as somebody who is breeding tomatoes or peppers or
right,
apples or,
not at all.
whatever. They can't go to the plant variety protection office and register a new strain because it's illegal. So there's very little incentive for breeders to do that work, grow out multiple generations and select and make sure that they stabilize the traits because at the end of the day, they're just gonna
You just get ripped off.
someone's gonna relabel it or just grow it and not give any royalties or anything like that. So It's a little bit different in like the whole system in cannabis is different because of this prohibition, because of the
Mhm.
status, the federal status it being illegal. There's no traditional pathways for real anything in cannabis. And so, yeah, I can see, so cloning is the easiest way to get around that, to get a stable next generation. But there's issues with that too.
Mhm.
You keep cutting a plant, you're going to get mutations that accumulate and things like that.
Absolutely.
I think tissue culture can help with that. If you've got a tissue culture bank where you can switch out and grow out a new mother from scratch, kind of,
Mhm.
to make sure that you've always got a backup for disease or something wipes out your whole grow. So I think tissue culture is like super helpful in that sense. And then, oh, like, I'm super interested in double haploids. That would be like your instantaneous, no variation because you've got the same copy of each allele. Right,
Mhm.
but I'm also of the understanding that it's not that easy to do.
Yeah, I'm not super familiar with that or the process of how to get to that point, but I mean, in theory, yeah, that's super intriguing, right? To just have a completely homozygous genome,
Mhm.
essentially, right? It's something I'll have to dig into because that's a really interesting question. And I don't have a clue how you methodologically how you would go about doing that.
But that's how those big agricultural companies, that's what they do. They don't spend multiple generations stabilizing genetics, they just go straight to the double haploid and boom, they've got it.
Mhm. It makes
Uh,
sense. But even then, like, I think we're kind of running into this thing of I've been thinking about this more from like a food perspective of like food crops, of just monocultures and how we're just kind of decimating biodiversity. Where it's like, if studies have shown us anything, it's that when we decrease biodiversity, that's almost always a net negative.
mhm,
And so, this idea of growing monocultures for everything just I mean, just from a biological kind of natural world perspective, that's is unnatural, right?
mhm.
So it's yeah, that's a whole
And
nother issue.
right, and it's like,
But
have we learned nothing? Pretty much every episode, I mentioned the gross Michael banana that went extinct because it was a monoculture and there was a disease and it killed all of
mhm,
them. So now we have this stupid Cavendish banana that tastes disgusting that I hate, even though banana flavored stuff is my favorite flavor in the whole world.
right, banana's so good. Dude, there's a special banana that's only grown in Hawaii. I can't remember the name of it offhand, but like you're not allowed to take any like from the islands to the mainland or anything. But it's like this super specialized banana that's supposed to just be amazing.
I have 6 banana trees. They're blue
Okay,
they're
sweet.
they're blue Javas, and we have them
Okay,
outside, spring we bring them inside probably around October. So I have giant banana trees in my bedroom because I don't want them to
cool.
die, but they're the blue Java ones, so they're blue, they're kind of like that jade blue color,
Mhm.
and then when they get really ripe, they taste like vanilla ice cream, or so I hear. Yeah.
That sounds heavenly.
I
Like
know.
vanilla, I would say vanilla, banana, and orange are probably some of my most favorite flavors.
Really?
And so, like, yeah, anything vanilla and banana. That was, yeah. So I always wanted to breed a strain and I wanted to call it Orange Power. And that would be more like an ode to Oklahoma State because that's what gets yelled at football games and stuff. This call and response from the crowd. But Orange Power, and it tastes like of creamy vanilla orange. And then the high on it would be like a kind of like a flow almost of like, fuck yeah, let's go do something. Go to a football game, go disc golf, go do something, hike, whatever. But yeah, I wanted to breed one called Orange Power, and maybe one day I'll do that, but I have not made any progress on that so far.
We'll get there. Yeah, oh, you were talking like a long time ago, you were talking about like using computing power and stuff. And I was thinking about TrueCut. We now have a universe. We've got, 80 something samples in there and we're we've got cookies collection, we're getting a Brothers Grimm collection in there. We've got some land races in there. Brian is that's what he does, is AI,
Mhm.
mathematics, big computing stuff, machine learning. which is nothing that I do.
Mhm.
But he expects to eventually be able to predict breeding outcomes based on the parental genes. So if you are looking for this, then these are the two things you want to breed together. Or if I breed these two things together, then I will very likely get this outcome, which I think
Mhm.
is super interesting. I don't know how that's all going to work, but that sounds
Yeah.
like that would be right up your alley.
Yeah, that's super interesting. So instantly questions I have. I think you had mentioned one time before, like in order to do the true cut, you also have to have the parents as well.
If it's something that's already been out there, if you stepped forward and you said, I am the original breeder of Blue Dream, and
Hmm.
here's my Blue Dream sample, we would have to say, prove it. And
Okay.
if you had the parents and we could determine that, yes, these are the parents of this, of Blue Dream, then we could then potentially afford you protection, say you're the original breeder, you are the owner of Blue Dream. So, like Laura at Brothers Grimm, right? Mr. Soul, Rick Campanella, he is the creator of Cinderella 99. It's been out there and in the
Mm.
market for a long time now. And I was like, do you have the parents of Cinderella 99? And she's like, oh yeah. And I'm like, then we can afford that protection to Rick because you have the parents. It's been in your hands as Cinderella 99 for however many years. And so yeah, we can definitely certify that under you.
Nice. I just smoked some Cindy 99 a few weeks ago.
What you're doing is you're bringing forth all the things needed to empower the supply chain.
What we're talking about is genetic certification, but really at the center of what we're trying to do is market maturity. Well, just cannabis growers and breeders are facing problems that they don't have the same answers that traditional linear agriculture has.
How do you get people to come in that have the desire to do something interesting and bring value and not get screwed?
I've kind of been a bit of a hermit when it comes to socializing and cannabis and all of that stuff. I've kind of, yeah, just stay in my lab and be quiet and do science and nerdy stuff. But I definitely need to get out there and kind of socialize and mingle a little more.
You don't
Yeah.
have to.
Well, like I haven't even gone to like a conference or like a vendor show or like, NJ BizCon or something like that. Like I haven't been able to go to any of those yet, but I'm hoping in the very near future to do that.
My
Okay.
favorite conference is MJ Unpacked by far. And
Is
and
that the
they
one up by you?
yeah, so
Okay.
Atlantic City and St. Louis, Missouri, and then I think they added a third one, and I can't remember where that is. But yeah, that's by far my favorite. And then there's a few others. Like I think that Can Med is probably pretty good. And actually, like I do have a lot of friends that go to Can Med, and I think it would be really fun to just I went to the Sunny Niagara conference for fun for my birthday, gave myself a birthday present, went and hung out with my friends. Daniela says I'm weird for going to a conference for my birthday. But it was really fun. I got to hang out with there are some really, really cool people in the cannabis space that are
Yeah.
a lot of fun and super interesting. And you should definitely get out and get to a conference.
That would be awesome. That would be very
Yeah.
cool. So I wanted to ask your kind of just pick your brain a little bit, I guess. So while we were talking about gene edited and stuff like that, right?
Mhm.
And because of just partially of true cut and things of that nature, are there a lot of varieties or strains out there that cultivars? What word do you use?
I
Question.
use I use strain.
Okay.
And
All right.
the reason for that is that cultivars have a specific definition. They
Okay.
have to be unique, they have to be stable, they have to be I can only ever get 2 of the 3. There's 3 things
So a
that it
little more
has to be.
formal.
Yes,
Okay.
and it at cultivar is an actual, like, taxonomic status, right? And so it's kind of yeah, cannabis, it just isn't there. I mean, in most cases, it's just a cross of whatever, and then you clone it, and well, when it's around, but if all the mothers die, then it's gone.
Hmm.
Like, so I think strain is like could be a stepping stone to cultivar, but I
Okay.
don't think that most cannabis on the market are at that level. So that's why I
Gotcha.
prefer the term strain.
Okay. I say strain too, but I know some people have a bit of a contention with it. Just like, I mean, indica sativa, right? It's kind of, do we have just a better alternative? Not really, and at the end of the day, everyone experiences a particular product differently. Like that's why we run clinical trials because you never know how people are going to react to stuff. So,
I see indica and sativa as bucket terms. I don't like that people say indicas will make you sleepy and sativas will make you awake. That's not
yeah,
true for everybody. It may
right.
be true for some, but I don't like telling people how they're going to feel, especially with something like cannabis, which is so diverse. And then on top of that, you've got everybody's personal physiology, not only from person to person, but also day to day and
Mhm.
hour to hour, depending on what you've
Right.
eaten and how well you slept. And like all kinds of factors are going to play into that. So I think of indica and sativa as like they are different. Try them both, see which one you prefer.
Mhm.
And that you might like this one better at night. You might like this one better during the day.
Yeah.
So I see them as bucket categories, and as long as there's no effects tied to those words, then people
Mhm.
can make informed decisions and form their own preferences and have a word to say, I like indicas better. Why? Because I just do. Like, I like chocolate cake. Why? Because I like it better than vanilla cake. Like, there's no
Yeah.
reason why. Like,
Right. Because we
right.
can.
Yeah. So that's kind of the way I see it. And I also
Yeah.
think that the morphological differences are obviously the plastic traits that
Mhm.
the plant has adapted to different geological areas, different regions. Therefore, those plants, they look different. So there's an indica and a sativa type morphology, and they look different because they are adapting to different regions. But in those regions, there's also different pressures,
Mhm.
right? Environmental pressures, which means that they're going to produce different chemicals for different bugs, for different pathogens, for different UV rays, for different.
Absolutely.
And so it makes sense to me that those differences in whatever those plants are making, those chemicals would be different. And that very well could be what's lending to the indica sativa thing that so many people subscribe to.
Because,
So
like for at least back in the day, right, it was kind of indicas, like your Aptan Kushes and stuff. Those are going to be more kind of colder climate, shorter day length, right? Photo period, or not photo period, but just finishing time, flowering time.
shorter, better, more anthocyanins,
Mhm.
because antifreeze and sunscreen,
Yeah. And just, yeah, once you get to warmer climates, it's longer days and warmer, just
more
all the stuff,
chillax, more
humidity,
more
all kinds of
hanging
stuff.
out, being all lanky and
Right.
going with the breeze.
That's right. That's what I'm about, man. I'm like, I'm looking ready forward to the beach, honestly. Like,
Yeah.
ready for the beach,
All
but
right, then we got sidetracked. So you're gonna ask me
all
something.
good. But yeah, so this would be what I'm kind of curious about is I think it would be referred to as type 3. And so that would be no cannabinoids or like trace
That's type
amounts of.
5.
Oh, okay. So
So
2 is
1,
1 to 1,
yep, 3 is CBD
3 is
dominant,
CBD,
4 is CBG dominant,
CBG. Okay.
and type 5 is no cannabinoids.
Okay. So I guess in type 5s would be what I'm kind of somewhat really curious about. And that's this idea of, have you seen a lot of type 5? Is that kind of out? I know they exist from, I think Oregon CBD had something on their website, however many years ago, that they had basically cannabinoid free strains, but just had really high like terps. No.
Only time I've ever seen anybody do anything with a type 5 was a talk actually up at the SUNY Niagara conference where a lady, and I cannot remember her name, but she is a bug person, and the bugs decimated the, I want to say the type 1, ate the shit out of it and just didn't even
Huh.
touch the other one. And I think that's how, I'm pretty sure that's how it went, because I was like, wow, I don't know, I'll have to check on that. But it was really
Yeah.
interesting. Like, it was night and day. The bugs definitely had a preference.
Okay. Interesting, because yeah, I'm just thinking of, and this is more, this is kind of my synthetic biology mind working a little bit. A lot of terpenes, at least mass-produced terpenes, are initially derived from pinene that we get from harvesting pine trees for lumber and things of that. So it's really almost more like turpentine and stuff like that. It's like a byproduct that we get from the lumber industry. And so these industrial processes have been made, like chemical conversion of some terpenes is incredibly cheap. Like it requires high heat, high temperature, high pressure, stuff like that, kind of industrially. But again, these are all kind of, I don't want to say synthetic terpenes necessarily, but the idea of growing cannabis, not for cannabinoids, but for terpenes, and then extracting the terpenes out for consumer products, whatever, or it could be pesticides, right? Like limonene is a pretty good pesticide and stuff like that. And so by having no cannabinoids, you kind of get around the whole, being a love going hot or whatever. And so you can hopefully bypass that a little bit. But initially started as, could we take, like, say, a type 1 plant that's making 30% THC? Could we then do gene editing to deactivate one of the cannabinoid biosynthesis genes? I think I was looking at the alditolic acid synthase. Can't remember.
Mhm.
It was kind of upstream of everything. But if you turn that off, how much of that 30% THC could you then potentially funnel into making terps? So could
Hmm.
you actually push it a little higher by not making any cannabinoids at all? And I know that metabolic engineering doesn't always work like that, right? If you shut off one kind of valve, if you will, that doesn't necessarily mean it's going to go the way you want it to. But
Right.
rather, I think it would be an interesting place to start kind of poking and probing, like what does happen metabolically? What happens to the chemical profile of the plant when we start shutting things off, if you will?
I don't know, and I don't know enough about the pathways. Like, I've never actually, sat down and studied the biosynthetic pathways. That might actually. I'm trying to think who would be a better person to ask about that, but I don't know. I feel like if you're gonna shut something off, you close one door, another one's gonna
Mhm.
open. I
Right.
don't, and I feel like often in science, you have a theory and it doesn't do what you think it's gonna do.
Right. That's pretty common, I think.
Yeah,
For
and I
sure.
don't know why you want to jack up the terpenes. I mean, other than extracting. So I was
Mhm.
talking to Shauna Reiki yesterday from True Terpenes.
Mhm.
And so they do a lot of, I mean, they make terpenes, right? So they have, I think she said something like 120 or 150 raw ingredients that they get their terpenes from. They are botanical terpenes mostly, but they also have a line that are from hemp. So
Okay.
they're cannabis derived terpenes, and they are a little bit different because yeah, like when you extract these terpenes, other stuff is gonna come along with
Mhm.
that, and so the cannabis-derived terpenes are a little bit more cannabisy than the
Right.
botanically derived ones, and so they have, like, all of these different things, and I feel like it's one of those things where you're kinda doing science for science sake when there's a lot more easy path to go down to do the same thing.
For sure. I think it's more niche, right? Cannabis derived terpenes, because you think about making distillates and then people adding the terps back into
Yeah.
it and stuff like that. So I don't know how much of a market there is for it, but
He said it's more difficult to get the terpenes from the cannabis plant than it is to get it from other sources, which is why they
that
do
makes
it.
sense.
And so products with cannabis derived terpenes will be more expensive because the process is a lot more difficult.
Okay. That makes sense. All right. Yeah, I think at one point I had the idea of using like cannabis derived terps to make like sparkling water.
Yeah.
Like if you really just love the flavor of cannabis and wanted some sparkling water.
There's a few products out there that are very cannabisy. Like you
Okay.
can definitely taste the cannabis. There's a brand called Kalo, Kalo, and it's actually
Okay.
here in New Jersey. And they have, their hemp beverages. So they've got CBD in them, but they've got great flavors, but there is a definite cannabis taste to them as well.
Okay.
And when
That's
we
pretty
say cannabis, we're talking about the genus, not
right.
not marijuana,
Right. It's
because
so dumb.
it is so dumb.
So dumb. Like, it's cannabis, period. Like, I don't know. I think all these kind of redefining lines and rules and stuff, I don't know, it gets a little unnecessary in my opinion. I mean, I get it. THC messes you up. It can. So I understand it. But as far as all the rules, I mean, at the end of the day, I think it all comes back to just like tobacco and alcohol are just kind of pissed off. Like you look at drinking rates among Gen Z and stuff, and it's just like absolutely plummeted. I
Oh,
personally have always been, I would much rather smoke than drink any given day.
I went through this whole thing when I was writing the NIDA paper, which is, I wanted to look at the genetic spectrum of, wild hemp, cultivated hemp, CBD types, indica, sativa, hybrid, all the things. And then I had the 2 samples from the National Institute of Drug Abuse out of the University of Mississippi.
Okay.
And when I was writing that paper, I went through a whole thing. I'm like, all right, what are we going to call these things? Like they call it research grade marijuana. People hate the word marijuana. Okay. So hemp is hemp, right? So we know what hemp is. It's below 0.3% THC. It's for, fiber, CBD, whatever. What do we call all the stuff that isn't hemp? Do we call it non-hemp cannabis? Do we call it, and that's a mouthful. Do we call it drug type cannabis? Can't call it drug type cannabis because drug, and also CBD is a drug. It's actually a FDA approved drug. So we
Right.
can't call it drug type cannabis. So then we could call it, there was another, we could call it type 3 or type 2, 3,
Mm.
2 and 3 cannabis. And I was like, that's stupid as well. And I came to the conclusion that we don't have a good word. If you're one of those people that doesn't want to use the word marijuana, we don't have another word that accurately
Right.
describes everything that isn't hemp.
That's
Right?
fair.
If
Yeah.
we call that cannabis, now we're saying that hemp isn't cannabis, and hemp is cannabis. So it's like, and I get why people don't want to say marijuana because of the whole history of the word and how it was used. But the word was in use long before that. It was in the Mexican pharmacopoeia. It has a specific definition, which is the flowering tops of the female cannabis plant that is used and prepared specifically for smoking. And I'm like, that's exactly what marijuana is, right? And so, but I went through this whole thing in my brain, like, let's just do drug type cannabis. Okay, no. Let's just do marijuana. No, like it was just
Yeah,
so
I can
annoying.
imagine. Because even like trying to keep all the different types straight.
Yeah.
So it's not necessarily an easy problem to solve with just a couple words, I don't think.
We ended up going with drug type, hemp type, and NIDA.
Okay.
So we ended up doing drug type. But again, that's not right.
Well, it worked in the situation for right, just within the confines, the scope of the paper.
But yeah, like I just, this language gets so fuzzy because people want to step around or tiptoe around perfectly good words that have actual definitions. Because I don't know,
Yeah.
and the whole sativa indica thing, at first it was like, oh, THC is the thing. Then it's like, oh, indica and sativa is the thing. Now we're like, oh, terpenes is the thing. Terpenes isn't the thing either. We're finding out that, these almost every strain has, I think it was Abstracts looked at 20 different samples of different strains. All of them had limonene in them and they all smelled different. Like it wasn't, and they weren't citrusy like we think they are. Right. So I'm like, we just keep replacing one flawed system with another flawed system.
Agree. Yeah, we do. And it's, I don't know. Maybe one day we'll get around all of that, but we'll see. Because even like this whole, I mean, they've been saying Schedule 3 for a while now, and even with the EO, it's still, I think, nothing's happened to my knowledge thus far. So I don't know. I don't think Schedule 3 is necessarily the answer. I think DRAG is a better answer, but I
think everybody thinks that it's just not.
like at the end of the day, it's a plant, right? And I understand like some plants are a little more intense than others, right? Opium, things of that nature, but
I don't like that they want to regulate the seeds based on what the parents are. We don't.
that's completely unrealistic too.
You can buy poppy seeds. That's fine. You can grow poppies. You can, if you want, milk your poppies. And I don't, but we're going to go after cannabis seeds. I think that's so stupid.
I remember reading back in the day about one of the reasons for outlawing marijuana was to keep immigrant populations out of the city. They didn't want, like, right? I think it was like Chinese immigrants or something like that, workers, and they would smoke it, and they didn't want the immigrants in the city, like, limits, if you will. And so they just made marijuana illegal. And so they just would stay outside of town. So it's kind of, there's certainly a loaded history behind it.
Mhm.
And
Well, and we
it's
have to
weird.
remember that Schedule 3 is not necessarily the end of the road. It might just be a stepping stone
Right.
to complete
Right.
descheduling, which is what I think everybody wants.
I agree. And if you look at kind of what is it, the addiction rates for different drugs, and you have like heroin and stuff like that, it's just off the chart, so to speak. But if you look at stuff like cannabis or hallucinogens and stuff like that, the addiction risk is like next to nothing. Yeah, we're like completely demonizing these things like they're the worst things you can come across. So it doesn't make sense, honestly, but
It doesn't.
that's the system we live in at the moment. So
Super annoying.
agree. I completely agree.
All right. Chad, do you have anything else that you want to touch on or talk about or anything?
I could tell a story from undergrad. You mentioned the plant systematics course, and I had to take a field botany course during undergrad. So in that course, we had 4 Saturday field trips. They were like 14, 16 hours long. So get up, get on a bus like 6 a.m., and then go to one corner of Oklahoma. And so we were going to the northwest part of Oklahoma. And I didn't pack any lunch, but what I did pack was I made a big batch of special cookies. Um, and that's all I took with me. And like an idiot, stupid college kid. And so we're doing the trip and then we're going and I'm just hungry. So I just start eating cookies and I'm just like, whatever, I'll just eat a few. Well, I end up eating more than a few. And by the time we stop for lunch, I am just lit. I am super high. And I started to get super anxious and nervous that everybody else knew I was high because there was one other guy in the class who obviously smoked while we made our pit stops at various places to go botanize. We stopped at lunch at a park, and I sit at the table furthest away from everybody to just get away from everybody. And then the professor and the TAs come sit at that table as well. And what was funny is the professor, he was from the Netherlands, and we were sitting at the table and he was wearing a shirt that said Oklahoma. And then one of the TAs said, "Oh, I should have worn my Netherlands shirt." And so in my mind, I'm thinking, oh God, they know that I'm just completely lit right now. And a few minutes go by, trying not to make eye contact with anybody. And one of the TAs, Mary, she was like, "Chad," and she's like hollering at my team, just like, "What's up?" And she was like, "Did you do that on purpose?" And I'm just like, "What are you talking about?" And she was like, "You have a praying mantis on your head." And sure enough, I had a praying mantis that had somehow just crawled and was just chilling on top of my head. And I had no idea. And so I was just like, got it off or whatever, then got up and walked away. But yeah, it was just that was my edibles mistake, I guess you could say, on a college field trip. And I thought I was so busted, but yeah, no one ever said anything. But
Oh, they know. Those plant
oh,
folks,
I'm
they
sure they
know.
do.
They know. Our plant botany class was Mitt's class again, and he would do it in the summer, and it was a week-long camping trip.
Oh, wow.
And yeah, with undergrads and grad students, and it was very strict. No weed, no alcohol. But every year, there's always one.
Yes. Gotta
There's
keep it
always one.
interesting.
Yeah.
It is
So
college. I mean,
yeah. All right. So anyway, I need to, close this up because I got a dog that's hungry and some
sounds
feral
good.
cats that I feed. If people have questions about synthetic biology or any of the things that we talked about, you're on LinkedIn
Yeah.
and you're on TikTok too. You make amazing videos. I love your
Thank
videos.
you.
So follow Chad on TikTok and hit you up on LinkedIn if they've got questions, right?
Yeah, I would say LinkedIn's probably the best. That's my more, I guess you could say, professional front, if you will. TikTok, I kind of random, personal,
Like all kinds of
whatever.
stuff.
It's kind of the whole gamut, really. But it's been interesting. I've only been on TikTok literally for less than a year. I was vehemently against social media forever. And then really, right, I think that's kind of that neurodivergence thing. It's like this idea of I can just not show myself, express myself, whatever. I can just sit down and do the work, and my brilliance will do what it needs to do. And not quite. It's like being a hermit is, it does
Yeah.
have its drawbacks. So
No,
time to be a little more social.
yeah, I like the variety of your TikToks, and I do really enjoy the sciencey ones. You've got a really good way of explaining complex topics where I'm like, oh,
Thank you.
I understand that now. Because there's a lot of stuff about plants that I don't understand, and I like to learn from people who know more than me. It's been great conversation. Thank you so much for coming on my Flora Fridays.
Definitely. Thanks for having me.
Yeah, you're welcome. And we will be talking soon, I'm sure.
Definitely.
All right. Have a good day. Have a good weekend.
You too. Thanks.
All right. Thanks. Bye.